Welcome, students, to today's lecture on cardiac muscle histology. In this detailed script, we will delve into the intricate structure and function of cardiac muscle, as well as various histopathological features and abnormalities associated with cardiac diseases. So let's begin. Cardiac muscle, also known as myocardium, is a specialized type of muscle found only in the walls of the heart. It exhibits unique characteristics that allow it to contract rhythmically and efficiently pump blood throughout the body. Unlike skeletal muscle, cardiac muscle cells, or cardiomyocytes, are interconnected, forming a network that ensures synchronous contraction of the heart chambers. Cardiomyocytes are cylindrical cells with a single centrally located nucleus. They are rich in mitochondria, essential for generating energy required for sustained muscle contraction. The cytoplasm of cardiomyocytes contains myofibrils, composed of contractile units called sarcomeres. These sarcomeres contain thick myosin filaments and thin actin filaments, responsible for muscle contraction. Intercalated discs are specialized structures that join adjacent cardiomyocytes. They consist of three components: desmosomes, adherens junctions, and gap junctions. Desmosomes provide mechanical strength, adherens junctions mediate cell-to-cell adhesion, and gap junctions allow for direct electrical coupling between cells, enabling coordinated contraction. Excitation-contraction coupling refers to the process by which electrical excitation leads to muscle contraction. In cardiac muscle, it involves the depolarization of cardiomyocytes through the spread of action potentials initiated by pacemaker cells in the sinoatrial node. Depolarization triggers the release of calcium ions from the sarcoplasmic reticulum, leading to the sliding of myofilaments and muscle contraction. The cardiac action potential consists of several phases: depolarization, plateau, and repolarization. Depolarization is initiated by the influx of sodium ions through voltage-gated channels. The plateau phase is characterized by the influx of calcium ions, which prolongs the action potential and ensures sufficient time for ventricular contraction. Repolarization occurs due to the efflux of potassium ions. The cardiac conduction system maintains the coordinated electrical impulses that regulate cardiac rhythm. It consists of the sinoatrial node, atrioventricular node, bundle of His, and Purkinje fibers. The sinoatrial node initiates each heartbeat, while the atrioventricular node delays the electrical impulse to allow for ventricular filling. The bundle of His and Purkinje fibers rapidly transmit the electrical signal throughout the ventricles. The cardiac cycle encompasses one complete heartbeat and involves the contraction, systole, and relaxation, diastole, of the heart chambers. During systole, the myocardium contracts to expel blood, while diastole allows for filling of the chambers. Histologically, cardiac muscle exhibits distinct features such as striations, centrally located nuclei, and intercalated discs. Cardiac hypertrophy refers to an increase in the size of cardiomyocytes, often in response to chronic stress or pathological conditions such as hypertension or valvular heart disease. Histopathologically, hypertrophic cardiomyocytes display enlarged nuclei, increased protein synthesis, and disorganization of sarcomeric structures. These changes can lead to impaired cardiac function and eventually heart failure. Cardiac fibrosis involves the deposition of excessive collagen and other extracellular matrix components in the myocardium. This process is commonly seen in various cardiac diseases, such as myocardial infarction or chronic inflammation. Histologically, fibrotic areas appear as regions of dense, collagen-rich connective tissue. The presence of fibrosis disrupts normal myocardial architecture, impairs electrical conduction, and contributes to cardiac dysfunction. Unlike skeletal muscle, which has a limited regenerative capacity, cardiac muscle has minimal regenerative potential. However, recent research has shown promising findings regarding cardiac regeneration. Stem cells, tissue engineering, and other therapeutic approaches aim to restore damaged myocardium and improve cardiac function. Histological evaluation of regenerated cardiac tissue focuses on assessing the restoration of cardiomyocyte architecture and vascularization. Various cardiac diseases display distinct histological abnormalities. For example, myocardial infarction exhibits necrotic cardiomyocytes, infiltrating inflammatory cells, and scar formation. Dilated cardiomyopathy is characterized by enlarged and irregularly shaped cardiomyocytes with thinning of the myocardial wall. Understanding these histopathological features provides important diagnostic and prognostic information. Comparative cardiac muscle histology explores the differences and similarities in the structure and function of cardiac muscle among different species. Comparative studies allow researchers to gain insights into evolutionary adaptations and identify potential therapeutic targets for human cardiac diseases. In conclusion, the study of cardiac muscle histology provides essential knowledge for understanding heart function, diagnosing cardiac diseases, and developing therapeutic strategies. From the microscopic structure of cardiomyocytes to the complexity of the cardiac conduction system, each aspect plays a crucial role in maintaining the integrity and functionality of the heart. By exploring histopathological features and comparative studies, we can further unravel the mysteries of cardiac muscle histology and pave the way for advancements in cardiovascular medicine.